أيوة دكتورة صوتك واضح.
اوكي، بس الظاهر دكتورة نورا طلعت، بس استناها. دقيقة شوي. ايوه
ألو.
هذا هي. ايوه تفضلي دكتورة نورا.
معلش الكونكشن عندي. Okay, we will start. Welcome everyone. My name is Dr. Noura Al-Saleh, consultant medical genetics metabolic medicine. I'll be your moderator today. I'm happy to present our talker today, Professor Amal Hashim, who's a consultant in medical genetics and metabolic medicine. She's the head division of the medical genetic and metabolic medicine at Prince Sultan Military Medical City. Professor Amal Hashim awarded her bachelor degree at King Saud University, followed by her pediatric training in Prince Sultan Military Medical City, where she got her Saudi board in pediatrics and Arab board in health specialty, followed by her fellowship training in genetics and metabolic medicine at King Faisal Specialist Hospital and Research Center. Professor Amal is also adjunct professor at Al Faisal University. She has many international and national talks and also well-known publication, around 90 published articles in well-known national and international journals, and around more than 30 international presentations. So our topic today will be talking more, going with the theme of COVID-19. So she will be talking about our genes may explain the outcome of COVID-19. And I will leave the stage for Professor Amal, but just one for everybody. If you have the Q&A or questions you have, please put it in the Q&A tab, not in the chat, so it'll be all in one place and will be easier to follow. Thank you very much, and you can go ahead, Professor Amal.
Thank you very much, Dr. Noura, for your nice introduction. Good morning, everybody. It's very nice day today, and it's my pleasure to present the hospital lecture, hospital grand round lecture. And as Dr. Noura said, with the COVID era, and everybody's talking about COVID, so I suppose that also we have to contribute to this from our division. By the way, this is our new logo for the division for metabolic and genetic, which I like it a lot. And I have to explain it to you because, you know, this is the DNA, this double strand DNA, and we put we incorporate the molecular because we are responsible for the metabolic section as well. And here's the, you know, the father and mother and kids. So we try to incorporate all in one logo. I'm just a fan with our logo and I want to explain it to everybody. So let's talk about what I'm going to talk about today. It's about the era of personalized medicine. And this is a very important thing to start with, as I want to explain how can our gene explain the reaction to any disease or a drug. And when you see some people who had different response to a drug or treatment for a patient, and between 2 or 3 patients, that's because sometimes their gene actually is the one which is responsible for this, and they call it now personalized medicine. It is not new for this year, of course, but it is improving year after year. And then we'll start, of course, about to 2020, where all started this year. We'll talk about genetic gateway to COVID infection, and then our conclusion. Doctors have always recognized that every patient is unique, and doctors have always tried to tailor their treatment as best they can to individual. You can match a blood transfusion to a blood type. That was important discovery. What if matching a cancer cure to our genetic code was just as easy as a standard? What if figuring out the right dose of medicine was as simple as taking a temperature? And this is the dream that it will be by the personalized medicine. And just to make it very easy, it's very simple. Just imagine that your genes makes that you have 3 receptors. 3 receptors. Let's put this by the Laser point, yeah. So you have 3 receptors for specific drugs, and you give the 3 doses or this amount of doses for the drug. So you have here that this drug is effective and everything is fine. Just imagine that your gene has make the receptors only 2 receptors for this specific drug. And again, we are giving the same dose of drug as we used to do. We are giving the same dose for each drug for every patient, sometimes not in pediatric, but mostly in adult, that we are using it. So just imagine that this patient has only 2 receptors and we are giving him extra dose and he will have toxicity. And despite that, he had the same dose that the other people had. Another patient or another person, he may have, because of his genes, he have 4 receptors. So when you give him the standard dose, he will have only 3, which is much for him, and this will be not effective. And this is simply what we talk about personalized medicine and how the medicine should be tailored according to our genes. And year by year, we are reaching this, inshallah. It's not anymore one size fits all. No, sometimes it's not. We are trying, but it's not. So let's see how it's working, because it's all start with our gene, it starts with our chromosome. So this is the double-strand DNA, and this is double-strand, and here we have the nucleotide. If you open your genome, it is full of nucleotide, and this is the sequence. Of course, this is very small part of our genome. And as you see, it is A, T, C, A, T, and go on. Every 3 codon, it will give us, every 3 nucleotide will give us a codon. And of course, all of us, we are sharing the same genome. You know how much we are different between each individual in our genome? How much do you think we shared our genome? It is what they say that it is, we are sharing from the genome, we are sharing 99.9%. This is what we share in our genome, and it's only 1% or 0.1%, sorry, it's 0.1%, it is those 0.1%, let me put it in, sorry, yeah, which the people will be different between population. And what is this percent which we will different? This, what we call it sometimes, some of the difference we have is they call it the SNPs. What is the SNPs? And we will talk about it a lot. You will see me talking about SNPs many times. So SNPs, which is singular nucleotide polymorphism. This nucleotide is supposed to be C and in front of it G, but some people, they will have a T and in front of it's A. And this change is not abnormal. This is normal. This is polymorphism. This is change between population. So single nucleotide polymorphism are the most common type of genetic variation in a human. Shall those affect our gene? Let's see. A single nucleotide is sometimes replaced in the gene sequence, and different single nucleotide polymorphism expression may modify the drug therapeutic response to adverse effect coincidence. So those changes occur through the whole our genome, and it's about every 1,000 bases. So, and it is under intense study from the pharmaceutical companies, because they need to know what's going with other genes and why people are responding differently between some people to other. And just imagine that you can collect those population who have special SNP and those who have another SNPs, and every population, they have the same different of the SNPs. And then you see, oh, those people, this population have severe allergy, for example, to penicillin. And those, you have to increase the dose of penicillin, because usually if you give them their standard dose, they will not respond. So this is the characterization of SNP may help to identify the subset of individual at risk of specific disease. It also may predict drug response and adverse reaction. So some people, they may have drug toxic, but not beneficial. Other people, they may have the drug not toxic, but beneficial. Some, they will know that the drug toxic, but beneficial, and it's not, or people that it's not toxic and not beneficial. So how to go from there? So this is what we call about the natural variation of our DNA, which play a role in our risk of getting or not getting certain disease. And here how I'm going to talk about the COVID and COVID in response to our genes. External factors such as environment, diet, exercise, along with the polymorphism, all of those can determine the individual risk for diseases. And based on patient individual genetic expression, biomarkers, DNA microarray, all can help to detect the best medication for the people. The old pharmacogenetic or old personalized medicine, we all know the severe drug reaction for certain drug. You know what is this? This is the severe drug reaction to carbamazepine tegretol. And they find that genetic variation may be associated with the form of Stevens-Johnson syndrome or toxic epidermal necrolysis. So this is one kind of pharmacogenetic, how we can change our disease or our treatment and tailor to our patient because of their gene. Because those population who had HLA-related variant, they may be more likely than other patients to have dangerous skin reaction. So testing this allele can reduce the frequency of this reaction. Of course, if you have population of or patient with this reaction, you have to check him because he may give it to his kids. So just I talk about how is the personalized medicine, and now we will talk about exams. Because are not exams, just we start exams these days, and it was just 1 year back when in December 2019, a novel severe acute respiratory syndrome, SARS, from a new coronavirus, and by the way, this is the 7th coronavirus, was recognized in the city of Wuhan in China. And rapidly it became an epidemic in China and spread to all the world. It was 1 year ago when the first patient was infected after he ate a bat or whatever, and then he emerged as an epidemic. WHO declared it as pandemic, and 4th of May, everything was closed after it, and spread has shown extreme geographic variation. It's January, it was here, then February it extended, and then in March it extended more. The reason behind the geographic variation is still not known. There exists significant variation in symptoms profile and severity also. It's not only geographic variation, also some population has different response to another. The first case in KCA in Saudi Arabia, it was in March. And after the approval, the transmission from animal to human, it was proven to have human to human. The virus later detected in the bronchoalveolar lavage, sputum, saliva, and throat, and nasopharyngeal swab. And from WHO, you can take this and update this map daily. This was taken before in May, and then suddenly the number of infected cases has been rising dramatically. Till we have this in our days, and as you see now, it is full everywhere. Sorry. So there are several factors and ways that propose to influence COVID-19 pathogen, and the most notable risk factor was believed to be age and other comorbidities like diabetes, obesity, cardiovascular, and cerebrovascular disease. The most widely anticipated pathogen mechanism was the cytokine storm. We'll talk about this, and we'll talk about the genetic variation and the immune function that make the disease altered between population. And this should be started by the host genetic background, our genetic background. It is well-known fact that host genetic background play a vital role to determine the susceptibility to viral infection. And this rule begin with your host recognize the viral particles. Some population, we find them that they can, and some gene make your body recognize the virus faster than the other gene, faster than the other population. Sometimes the presentation of the viral peptide to the host immune system may be different between people to people, and we'll see how this can be explained by our genes. Neutralization of the viral infection, this is another factor. Some other factors like comorbid condition or environmental variable like temperature, exposure to sunlight, all have important mediation rules. How scientists study this genetic correlation? As I showed you before, we need to know about SNPs, and there's another thing. Both something called candidate gene and something called genome-wide association study. And when you read it, you say they always write GWAS, genome-wide association study. Those two techniques can offer significant insight into the genetic basis of many infectious disease. Simply, you can have some population, target population, you find that they have specific phenotype, specific special things they have it than the other people they don't have. It could be worse reaction for COVID, it could be better reaction of COVID, it could be whatever you want. You just collect those population and put like other population, they have the same environment or the same exposure, but different reaction. And then we sequence all their DNA, and then they will identify the SNPs, or we talked about the SNPs before, and from there they can verify what is going on. So this study identified genetic loci and allelic polymorphism that determined in part genetic susceptibility to infection. Studies of viral and host genetic are critical for understanding the pathophysiology of the SARS with COVID manifestation that is different from individual to another. Some people, they have very minimal symptoms of SARS, and some people, they have really bad COVID that reach to death. And it was this response to any medication or response of gene to disease is what was well known since the HIV. And it was thought that, and it was found that some population, when they get the HIV, they will not have bad HIV. And some population, they will have severe bad HIV. So what's happening? They find that this is the HIV. And when it want to come to their cells, there is a gatekeeper. There is a door where the gatekeeper is the gene which is called CCR5. This is the gene. CCR5 is the gatekeeper for the HIV to go inside the cells. So CCR5 is a normal gene we all have this CCR5. Some people, they have deletion in the CCR5. Their gene is mutated. Their gene is abnormal and deleted. So this gatekeeper is not there. This door is not there. What will happen? What will happen that HIV cannot come inside the cell, and this patient will have resistance to HIV. So actually, this mutation make it good for him, and he will live because he will not be affected by HIV. This is the gene which, when it's mutated, they call it delta CCR5. So can we identify such association with SARS-CoV? Because COVID is the disease, SARS is the name of the virus, which could help guide the design of vaccine and therapeutic, as well as to prioritize our individual for the treatment, and we can find by them effective vaccine or antiviral therapy to develop it. So let's talk about the first genetic gateway keeper, which is ACE enzyme gene. The role of angiotensin converting enzyme 2 gene and entry of SARS-CoV was found in some research and analysis. And maybe all of you know that the infection and death rate in male and COVID is more than female. It is around 1.4 to 2 times more than female. And this was found in China, Germany, France, Italy, and India. I have to say something here before I continue. Some studies, and this what make some people get frustrated and angry, because when any research or project is done, some study, they will find a result. This result was done only in their lab. This should be replicated. This study should be replicated in another population. If you have the same replication in other population and the same study, you know that this disease or the results are good. So always when they told you, oh, we find something, but it was not replicated in another studies, it is okay. This is the science. And all of you, that we know that the science have to take time. Don't be frustrated with other people who say, why COVID every time is changing? People is changing, scientists is changing their mind. They are not changing their mind. We are collecting information. Just because now in this era, we have a lot of connective, a lot of social connection, that everything will go from population to population, and you just get frustrated from the fast news spread. So let's go back now for our ACE. So this study was replicated in China, Germany, France, Italy, and even India, and they find that the male are more common time exposure to female. Let's talk now about the ACE2 gene. This is the ACE2 gene, which is located in chromosome X. They find that the ACE2 has generally been found and more expressed in men than women. Now, what is going with it? The SARS-CoV virus would like to go to the human cell. They will use this ACE2. So the more the ACE2 is there, the more the viral will come. The more overexpression of this one, the more the viral will come. If we can stop this ACE2 or make something with it, we can manage with this virus. The ACE2 is primary cellular receptor for the SARS-CoV-2. Studies have begun to investigate the relationship between the ACE2 polymorphism that I talked to you about. If people had polymorphism in this one, what will happen to the virus? Would it be less permissible and it's not coming inside because the gatekeeper is not there? And sometimes we want to give something that affect this one. And if we affect this and we remove it, what will happen to the human body? You know that the ACE2 is important for the heart. So what it will be the adverse effect of this? A structural modeling study predicts that certain ACE2 variant may provide potential resistance to SARS-CoV. So some studies showed really, if you have modified this, you may have SARS-CoV resistance. And recent study from Europe and Asia identified two ACE2 variant. And when I say ACE2 variant, that means that there is two change in the gene of the patient. And those are the two change. And they find that they have lower binding affinity to the S protein, and the effect of the viral will be lower. And this have potential to alter the kinetic of viral binding and the internalization of it, and can explain the significant observed inter-individual variability in COVID-19 case sensitivity. So we talk about the ACE2. Okay, now ACE2, sorry. There is another gene, which is the TMPRSS2. And this gene, they find that it has protease function. Is it good for us or not? This gene will act with the virus. It will remove its spike or cleave the spike. The viral will go rapidly here. So if this will act good, the viral will come here faster. And let me remove all my sketching here so we can talk and draw another sketcher. So this is I'm going to talk about the regulation of the TMPRSS2 gene transcription and process to severe acute. Why some of them, they have severe acute SARS or COVID because of this gene. And this, you see this androgen receptors and chromosome and ACE2 gene, all are present in the X chromosome. So the androgen receptor here and the ACE2 receptor here. ACE2, and respectively, testosterone and dihydrotestosterone stimulate the androgen receptors. Okay, the activated androgen receptors regulate this one. It will be activated. It will regulate the transcription of the TMPRSS2. When it regulated, the SARS engaged here. Here it will regulate this one. It is here. So the SARS receptors use the spike. The SARS receptor, the spikes will be removed by this gene. If the spikes remove, it will go inside the cells. Okay, so now this gene, when it's activated, it is here, and it will remove the spikes, and the SARS will go inside. And of course, this is very simple way. So if I can do something that inhibits this gene, as what they say, serine protease inhibitor, like cametose, like camostate mesylate, can inhibit the TMPRSS2 and partially block the SARS-CoV spikes protein-driven entry. And this is another pharmacogenetic way and research place they are doing. As I told you, any study should be replicated, should be judged, should be find that it is better. I mean, if it's replicated or you have more patient with it, that you say that this is the more trusted to be really or good. So the research from Genomic Consortium, a global collaboration to study a critical illness, compared the genetic information from COVID patient, and this was just published last month or this month in Nature. They examine many patients, more than thousands patients, like 20,000 patients, from ICU patient and versus healthy volunteer, from where they took the blood from UK Biobank, Generation Scotland, and 100,000 genomes. And what they study the DNA and the variation between the really severe responsive, those patients in the ICU who got severe bad COVID, and those who had the healthy volunteers, what happened to their genes. And the team found key difference in 5 genes between them: IFNAR2, TYK, and those genes. And this partially explains why some people become desperately sick with COVID while others are not effective. And let's talk about each one simply and fast. Okay, I will just jump to conclusion, because with the conclusion, I put some results. They have concluded that critical illness in COVID is related to at least 2 biological mechanisms. It's important about the first one is your immunity, innate antiviral defense, which is known to be important early in the disease. As more as your innate antiviral defense is active, as more that you will recognize your enemy faster, and you can fight them faster and better. And those 2 genes were responsible: IFNAR2 and OAS gene. And when they find that if IFNAR2 gene, it will bind to interferon. You know the interferon. It will bind to the interferon, and when they bind to the interferon, it will make your patient better, and I mean the response will be good to COVID. But what's going on if there is rare loss of function? If this patient has rare loss of function of the mutation in this gene, it's associated with severe COVID and many other viral. Why? Because they don't have this gene. They have late response of their body to know that they got the COVID. If they are late يعني بدري عليه, you will not respond to COVID unless late, and this make it severe COVID infection. So this suggests that this finding suggests that administration of interferon may reduce the probability of critical illness. So that's fine. Okay, give them interferon and everything is fine. What happened later on? There is evidence that cannot distinguish when the illness start the treatment may be effective. So some people, when they give them the interferon, they respond. Some people, they did not respond. So exogenous interferon treatment didn't reduce mortality in hospitalized patients in large-scale clinical trial. And why this? And what does this mean? This suggests that this genetic effect may be mediated during the early phase of the disease when viral load is high. So it's again that if you recognize it early, if you test your patient early, for this they say we will do mass treatment, mass testing. If you test them early, you can give them interferon early. Some of them, they may have better outcome. Another important factor, we'll talk about the innate antiviral defense. This we talk about it. Another factor, which is host-driven inflammatory lung injury. We'll talk now about the host-driven inflammatory lung injury factors, which is the key mechanism of the life-threatening late COVID-19. And those genes, they find those 3 genes associated with it. So again, now you have genes that can cause inflammatory lung injury. So go and suppress this inflammation, give them anti-inflammatory drugs. So JAK inhibitor, baricitinib are known to reduce the activity of TYK2 gene and hence may have potential management in patients with COVID. Indeed, FDA has recently issued an emergency use for the Bastanib and combination with remdesivir for the treatment of COVID in hospitalized patient. Okay. Again, people, please, I would like to remind you, whoever participates, you are most welcome to our lecture, and please write your name clearly in the chat, or write it clearly in your name, so postgraduate can use it to write your certificate, and you'll have your free CME. So Basma, you will like me because I kept reminding them. The third important gene is the OAS1, and it is really interesting gene. The OAS1 variant, this gene is important. Any variant with it is implicated in susceptibility to SARS-CoV-1 candidate gene. So, and this was a study in Vietnam and China. What is going on? This sketch is from another OAS3 gene, but it is the same explanation, okay? So this is the virus. This is the double-strand RNA virus. This is the RNA virus. And when it's come to inside the cell, it will activate in the cytoplasm. When it affects the cytoplasm, this OAS will detect the virus, okay? When OAS detect the virus, that this virus come inside the cell, it will synthesize a molecule. This one will synthesize a molecule called 25A, because simply we'll call it 25A. This molecule 25A has activity that it will bind and release something called RNAL. RNAL, which homodiisomerize and target the host and viral ssRNA. So this RNAL will act on this virus, the double strand and single strand RNA. What will cause? It will stop the viral replication. Okay, again, I would like to say it again, because it's really nice, right? Nice gene, that what it's going on. This gene will recognize your double-strand RNA virus. This is the double-strand RNA virus. When it's come inside, it will activate something with another RNA, which will stop replication of the virus. So therapeutic gene, or the use of this gene, provide the potential therapeutic target. Endogenous phosphodiesterase 2 activity, it will degrade the host antiviral mediated 2,5A. The therapeutic PDE12 inhibitor are available. So we have something called PDE12 inhibitors, which is available and augment this thing, and it will stop the viral from replication. And this is why those population who have some variant of this gene, they are protected against COVID gene. Against COVID infection and against COVID SARS-CoV virus. So we talked about two gene. Now I would go to talk about one theory about the ABO locus, and we have here our hematologist with us. The ABO locus was previously associated with COVID-19, and there's theory that the blood group A has worse outcome, and they will have faster infection than blood group O. And this was done in a study in 1,077 patients were infected with SARS-CoV-2, compared with 3,690 for normal people from Wuhan city, and replicated again to 23,000 people from another Shenzhen city, suggests that blood group A has significant high risk for COVID-19, while blood group O had lower risk. But the the latest study in Nature this month couldn't have the same result. They couldn't have the same result of things. But they find that there is single closed genome-wide significant at this locus in combined meta-analysis that suggests that this variant in ABO locus may be associated with susceptibility to COVID-19, but not critical illness. It may suggest that they have people with group A, they are susceptible to COVID-19, but not to be worse outcome because they have blood group A not all. And what is the theory or the explanation? It's because that blood group O has the anti-A antibody in the serum. The blood group A, they don't have this anti-A antibody. And this anti-A antibody can inhibit, you see this anti-A antibody, you see this is the human cells, this is the ACE and other adherent process, adherent cells. So the virus will come here and come inside. No, the anti-A antibody will stop it from coming inside. So this is the benefit of anti-A antibody. And this is the explanation why some of the AB or O group have less susceptibility to have the viral than the others. So we talked the first gateway, and now we are going to talk about the second genetic gateway to COVID infection, HLA. And all of you know the HLA molecule are important immune regulatory mechanism. It's encoded in our body by the MHC gene, which is major histocompatibility complex. HLA gene exhibit extreme diversity and have several thousand reported polymorphism. It is well known, documented that genetic difference in HLA genes account for individual variation to immune response against pathogen. You know that we have a lot of change in HLA of people who will have it. So whether distinct HLA allele distribution in various populations confer the protection of SARS-CoV is a topic debate and discussion. Some of risk of HLA allele reported for SARS-CoV-1 include those HLA numbers. I'm not going to count them. But contrary to this, some study could not replicate this finding and reflect that any association between HLA and risk. But we have another interesting thing, that HLA gene may also affect phenotype associated with COVID. And all of you know that olfactory dysfunction has been reported to be clinical presentation to mild to moderate form of COVID-19. And most of the people who got the COVID, he had lost the smell and the taste. Most of them, not all of them, most of them. And the response was different. Some people for days only, and some people, the loss of the smell and taste from 3 days to 3 months. And it's really different. Some gene essential to individual olfactory perception, importantly, olfactory receptors, and those are HLA genes. So they find some polymorphism, which linked the extent of distribution of HLA gene and the olfactor. And again, for this, some people will have the last 3 months and other 3 days. The association between HLA and olfactory dysfunction has not been tested in COVID-19, but it would be interesting to examine the particle role in HLA and olfactory dysfunction. And it is really nice subject to study. So we talk about 2 gateway for genetic, and now I'm going to talk about the 3rd genetic gateway, which is the cytokine storm. It's becoming increasingly apparent that SARS-CoV-2 infection lead to cytokine storms and exacerbate inflammatory response. In recent study, COVID-19 patients admitted to ICU were found to have marked elevation in IL-2, interleukin-2, 7, 10, GCSF, and others. Another study demonstrated the higher level of interleukin-6 were correlated with a 22-time greater risk of respiratory failure to COVID-19 patient. Cytokine production depends on cascading immunological event. T lymphocytes are prolific produced of cytokine, and the developmental differentiation and activation of T lymphocyte production of cytokines are controlled by the genes that, as well as epigenetic. So because our gene can produce this interleukin, so again, if you have the interleukin more or if you have less interleukin, you will have, some people will have the cytokine storm or not. It is not only that we have a mutation in gene. It's not only that we have mutation in gene or metabolic disorder. Metabolic disorder, we call it inborn error of metabolism. Now they are talking about inborn error of immunology, inborn error of type of immunology and IFN, immunity, in patients with life-threatening COVID-19. And this was published in Science before 2 months or 3 months. They find that rare variant in some genes, especially in this gene, and another gene, And if those people had this variant, they will have loss of function in the gene. If they have loss of function of those gene, the patient will have this, they will have more severe SARS-CoV-2. Those genes are important for our immunity and interferon types. Inborn error of the TLR3 and IRF7 dependent type can underline life-threatening COVID pneumonia in patients with no prior severe infection. You see this thing, no prior severe infection. Some people who had no prior severe infection, they were completely well. When they get COVID-19, they have severe pneumonia. And maybe this can be explained by some variation in those two genes. Why those two genes? Because loss of function of them, they affect the immunity of the people. So really, the COVID and SARS-CoV-2 virus, COVID infection, it really make lockdown, it's make us upset from 2020, but we were in a post that we need this lockdown, we need to clear our mind, our whether everything become more clear, even our science, because we concentrate in something that can help not only in COVID, it can help in other genes, in other things. But the most important question, what is our next problem? We are trying to manage things, but unfortunately and sadly, we are managing things and we are still the same population who are dealing with the epigenetic. We are dealing with our life the same way we are doing before COVID. We thought that our life before COVID will be different than after COVID. We are more washing our hands, we are taking care more of our people, our elderly people, but sometimes we are not changing from inside. إن الله لا يغير ما بقوم حتى يغيروا ما بأنفسهم. We are not changing. We are still, some of the practice is still there, not only keeping things after us. This practice, which was not changed, it's also reflect other practice in our way of eating, our way of dealing with smoking, with other epigenetic factor. I'm talking since 8 o'clock. I'm talking about genetic, but many genetic factor, I talk about it now. It will be affect more with epigenetic. Epigenetics means the multifactorial, means that your life, your way of life, your way of smoking or diet or exercise or sleeping, or this is also important, and it may change not the gene, it may change the expression of the genes. So what is our next problem? Our next problem will not be another Chinese people who will eat a bat or whatever. It will be the same way of our thinking. So my take-home message from these things, we talk about the role of ACE, that the other gene and specific candidate host gene and COVID-19 infection. Those genes may change the severity and disease outcome. We talk about the importance of performing a candidate gene epigenetic factor GWAS study across different racial and ethnic population in order to identify the gene and haplotypes associated with different factor of infection and clinical outcome, as well as vaccine response. And if you find the people who had response to vaccine different than other, sometimes not because of the vaccine itself, it's again because of his gene. Systemic genetic and biology study to understand different and interesting genetic effect on disease severity and outcome through the lens of immune response network theory. It's important to understand the viral genetic over time, not only our genetic, also the SARS-CoV-2 viral genetic, because they find that the sum of the virus genes is more severe, and the SARS-CoV viral itself has different, and every now and then it will change its genetic makeup, and through the geographic sum of the virus which is coming from like Iran is different than those which are coming from Italy, and particularly in the number and frequency of viral mutation and recombination event, that relationship to the viral effectivity and transmissibility, disease severity, clinical phenotype, viral burden, disease outcome, etc. This is very important to study more. It is important to understand the interaction and function effect of viral point mutation, recombination event on therapeutic monoclonal antibody, antiviral vaccine efficacy, and possible adverse event. So all these things, to understand all these things, we need to do like combination and collaborative study. And there's the COVID-19 host gene genetic initiation. You can go to it. And there's a many population there, many country they are there. And also Saudi Arabia is part of this, and it is part of it by the CACT. They have the Saudi Human Genome Program. Taiba University, they have another study about the genetic variation. Many studies in Saudi Arabia about the genetic variation or response to COVID in Saudi population intense. And inshallah, everything is fine, and everything after all lockdown, it starts to open, and we're coming with, we نعود بحذر, and we come with caution. And by this, thank you very much for bearing with me all the time. And thanks for every healthcare worker to contribute to saving life. Thank you for all the times when I remember March, April, May, how was the lockdown? How was the, musta'adeen and the volunteer work who take the medication to other people? Thanks for everybody. Thanks for who got the severe viral and who insist to work while just after they finish from their infection. Thanks for you all. And by this, I finish my lecture. Thanks, Thanks, Ella.
Thank you very much, Dr. Amal, and thank you for everybody who attended. Very interesting and well-rounded topic, and it came in a perfect time during this pandemic. I have a couple of questions here from the participants. First question is, as we know, aging modulates our genes. What specific genetic changes occur by aging in favor of severe infection that happen to elderly? So it's like basically asking, why do elderly have a more severe infection than a younger population? And there's another question that's similar, is asking why children having less severe pattern of the disease.
It is actually interesting question because is there any gene that can cause because the elder population has some worse, that we find that the elder population has worse response to COVID than the other. There are some studies about some genetic and some polymorphism, we can say, that can potentially increase COVID disease severity and aging population. And some studies so the answer is yes. There are some genes that they find it with older population. They may have the expression of the gene is different than the expression of this gene in the young population. And after this, they find that why those two populations has different expression and response to COVID. Some of those genes, some of those study, as I remember, was published in just in Journal of Genetics and last October, and BALF. One of those genes, and so there is the question. The answer is yes. There is overlap between host gene interaction with COVID and aged population, and some also they are talking about blood transcriptomic. So it is not only the gene that we have it in our body, and this is something I didn't talk about it. All of us, we have the same gene, but the transcription of the gene is different. So the translation of the gene will be different by age. And the transcription and the translation, after the changing, you have the same gene, but the protein which is coming from it is different from people to other people because of aging, because of other. And this is a transcriptomic. And there's, as I told you, there is GWAS, there is a TWAS, the transcriptome wide association study. And this is very good. This is very good question and very big, part of our issue to discuss and studies.
Thank you, Dr. Amal. Just to furthermore on this topic, There's a lot of papers out there talking about why elderly have a more severe reaction to COVID-19
Yes.
than young children. And overall, it's still not clear. But there
Yeah.
is a hypothesis that the aging immune system
Yes.
undergoes
Also.
immunosense, the T cell diversity alteration, and there's a chronic activation of the innate immune system known as inflammasome. Also changes in the epigenetics and dysregulation of the renal angiotensin system. So all these things, because they go under this chronic inflammation throughout their life, so their the T cell activation, these pathway immunology will have a more clear answer to that. But due to the chronicity, the exposure we're exposed throughout our life, our immune system is not as pure and active as a young child.
Yes.
Another question, which is the most popular question, probably you got asked this question on Twitter multiple times, is the vaccine safe, and does it give full protection?
Okay. That's, I was asked many times. Yeah. And then for this, I put in my status that I am not epidemiologist, I'm not infectious disease consultant, I am geneticist. So as the vaccine is effective, or what is the adverse effect, I have to know my limitations. So I will keep this for the ID people who I'm sure they talk and they are talking about it a lot. But if the other question, they ask me only if the vaccine is safe, isn't it, Dr. Noura? They didn't ask the other common question?
No, just if it's safe and does it give full protection?
Yes. Now, is it safe? I cannot answer yes or no. If the vaccine is there for myself, if the vaccine was approved by FDA, Saudi FDA, and as I trusted them many times, why not now? I will just stop trusting them. I don't think this is logic. And all of us, we are working in the hospitals for many years, and we know that every drug has adverse effect and whatever is there. So if this vaccine was there for and was tested on 37,000 population or people or more, or if we say 18,000, because this study was for two arms, people who got the vaccine and people who got the placebo, which is normal saline. So at least you have 20,000 people who got the vaccine, and it is apparent that it is safe for them. So why we have to predict or just think that it is not safe? Because it is a vaccine that we are doing vaccines, and the world, the scientists, they know the vaccine since many years. So why now we are talking about is it safe or not? Many drugs we are taking it, and many drug discovery every now and then. Why we are talking now about as if it's safe or not? As if we are not sure how we are doing our drugs. No, people, we know how to, that the pharmacies, the pharmacists, they know how to put A plus B and what is C. So why just now we are frustrated from other population? Is it safe in the long time or effective long time? What is shown now, it is effective. I'm not sure if it's everybody is taking, and we are seeing. I will take it as for sure, as I took influenza vaccine. But the question, which is always I'm asked about it, Dr. Noura, not if it's safe or not. Actually, does messenger RNA vaccine will change our DNA or not? And the answer is not. The messenger RNA will not change our genome. And if is there any technique that it will change our genome, we as geneticists will be happy to use it to help our people who are suffering from severe bad disease in our clinic. And simply the hematologist here, and they are suffering from the sickle cell disease, which is very small gene, the sickle cell anemia gene. It's very small, one mutation. It is known for all of them. If we find that the new gene, which is genetic gene therapy, is effective to change this gene, the same gene, not the genome. I'm talking about gene. We are talking about many years trying to change a gene, one gene, and we are happy that we can make it normal in a diseased patient. And it's not only one dose, it's many doses, many, a prolonged process. So it's not as simple that I will give vaccine and it will change my genome. It will not change my genome. it may change the expression of the messenger RNA vaccine. It is simply it will identify for you. Your body will know the sequence of the vaccine, the sequence of the virus. So it will start to fight it. It's like that you give a virus, so your body know this virus, know the gene of this virus, know the sequence of this virus, so it will make it immune against it, and it will make the antigen against it, and that's it.
Okay, thank you, Dr. Amal. They asked if it gives full protection. I don't think this is
our subject
for us as a geneticist, but you can ask our infectious disease colleagues. But as far as I read, it will protect from severe symptoms and signs, like the flu vaccine. And it has a limited time, like it's not going to be full protection once. It's like the influenza, you take it every year. So it may minimize the severity of the symptoms, but I'm not sure if it gives complete full protection. I have Dr. Ammari. I think he raised his hand. I don't know. Dr. Ammari?
I'm not sure if we can.
I don't know.
If we can, as Basma said. Yeah, I think Basma told me that the microphone are disabled, so he can. Dr. Ammari, if you can write your question and
question, you write it in the Q&A.
Dr. Nawaf is saying that the chat is disabled. Maybe Dr. Nawaf you can.
I don't know why it's disabled. But you can write and question and answer.
Okay, Dr. Anura, if the vaccine is there, are you going to take it?
Yes.
So just to make them more, comfortable that inshallah everything is fine.
As you explained, this type of vaccine has the least side effect. It's a sequencing of the S protein spikes. So it's not sequencing, it's not the full RNA of the virus. And you get the virus the same way. You get the messenger RNA of the virus, it replicates, it's spread throughout your body. So this is the same mechanism. So why this one will affect your genome and the virus didn't affect your genome? But this we give the sequencing of the S protein only, so it can be translated outside the nucleus. So it does not go near your DNA. It's in the cytoplasm where the translation occurs to make the protein, and then the body will recognize it and will make the antibodies against it. I think there's no more questions.
Maybe, I think Dr. Wala has a question.
Please type in the Q&A. I see hands being raised, but...
Yeah, she say, since the genetic makeup of individual is innate, how can we explain the less severe disease pattern in children?
Well,
and it's, this is the same when we talk about the elderly, as you say, there's some possible reason the SARS-CoV receptor, the ACE, is reduced in the respiratory tract of children, which we talk about it and associated with common cold. So it is may give some protection. The T helper immune response also protective in children. So many factors actually can explain this.
I don't know if is Basma still with us? Because Basma, the chat is disabled, so they can't write their name and their Saudi Council ID for the...
their Saudi Council number, they can write it after they will fill the evaluation.
The evaluation. Okay. So everybody, after we conclude, you will get an evaluation page that will pop up. You'll write your name and your information there.
Okay, thank you very much.
Thank you, everyone, and thank you for your valuable attention and questions. And hopefully, we add more knowledge and convincing and power to your knowledge, so you can relay it to your patients. And inshallah, we'll have an excellent response to this vaccine, bidnillah, and we'll be more and more able to travel and go back to our normal life as before.
Inshallah. Thank you very much, Dr. Noura. Thanks, Basma. So we'll just